This video is also going to be pretty short and sweet, but it's going to focus on endospores. So these really hardy, durable forms of a bacteria. Now, only some bacteria are able to form endospores when the environment gets kind of questionable. So when the environment turns unfavorable, it gets stressful. Maybe there's too many cells. Maybe there's not enough food. There's not enough space. Some bacteria are able to form endospores. And essentially, it's kind of like a hibernating bear. They go dormant. They're not doing any form of metabolism. They're not producing energy. They're just chilling and hanging out, waiting for the environment to get better. And it turns out endospores are really hard to get rid of. Okay, they're able to withstand extreme heat, extreme cold, a lot of chemicals, radiation. Okay, they're very, very durable. Okay, and endospores are actually why it's really hard for us to achieve sterilization of an environment. Okay, so when we say an environment is sterile, that means we've removed all forms of life, microbial, that is. And that's really hard to do because endospores are so hard to eliminate. Okay, and so only bacteria belonging to the genus Bacillus or the genus Clostridium are able to form endospores. So that means E. coli cannot form endospores. Staphylococcus aureus cannot form endospores. But Clostridium tetani, which causes tetanus, can. Clostridium botulinum, which causes botulism, can. Bacillus anthracis, which causes anthrax, can. Okay, now you don't need to go memorizing those examples, but when an organism belongs to the genus Bacillus or the genus Clostridium, only then can that organism form endospores. Okay, so most bacteria cannot, but it definitely aids in survival. It allows them to withstand stressful environments and then become metabolically active, what we call a vegetative cell. It goes back to a normal vegetative cell when the environment becomes more favorable. So in order to form an endospore, excuse me, the cell is able to detect that the environment is becoming questionable, and the organism is going to copy their DNA. You can see this down here in step 1. Again, trying to get my mouse to cooperate, sorry. So the organism is going to copy its DNA. Notice it starts to form a membrane around this DNA. Okay, it's kind of walling off this DNA. Then it's going to continue to form extra membranes or extra protection, okay, around this structure, which we're now calling the forespore. It's not quite an endospore, but it's getting there. So that forespore is going to form these additional membranes, and then it forms an additional protective cortex. Okay, so again, it's really just trying to protect that genetic material. So lastly, this protein coat is going to form around that cortex. And once all of these protective layers have been, built and are properly assembled, then the spore gets released from the current cell. And it's that spore that's just going to hang out and wait for things to get better. All right. That's it for endospores. We are going to go into more detail, more in the semester and in lab. So for now, just know that they're really durable. They're very hard to get rid of. Only certain bacteria are able to form these endospores. And then kind of just know the gist of sporulation, which is the process of forming an endospore.